When we try to use ultrasound for therapy in the brain, we have a major problem, and that's skull. And in this slide, you can see a CT of a skull, and you can see that the thickness and the density of the bone varies. And since the ultrasound speed depends on the density of the bone, the skull bone acts as a defocusing lens. And also the ultrasound attenuation and absorption in the bone is much, much higher, about 20 times higher than in the soft tissues. So we have double problem going through the skull. So if you use a standard focus transducer and try to go to the bone, the focus is diffused and we cannot get a sharp focal point. So we cannot overcome the enhanced energy absorption on the skull simply by focusing with standard approaches. So for a long time it was believed that brain is outside of what can be done with focused ultrasound non-invasively, that you had to remove the skull bone in order to be able to do surgery non-invasively in the brain. And it took us some time to figure out how to do the focusing to the skull, and the solution was to use phased array. And you have heard talks about the phased arrays, where we can control the amplitude and phase of each transducer element independently, and use the CT information from the skull to calculate how those waves are propagated into the skull, and then use this information to tailor the phase and amplitude such that we get a sharp focus to the skull non-invasively. And we had to measure all the skull properties dependence on the density also, and before we were able to do that, but about 10 years ago we were able to successfully demonstrate that in vitro human skulls. And that resulted in development of phased arrays, large hemispherical arrays, first in the lab and then with Inside Tech, a commercial product, where we utilize the whole available surface of the skull to get high gains in the brain. So it's a hemispherical array, and has about 1,000 elements in the current configurations to be able to compensate for the distortions to the skull. So this has been used clinically, and I'm going to describe only one example, and that's essential tremor. There are quite a few patients where they have tremors. For example, the hands are shaking so much that they cannot eat or drink easily. They need to have support or write. And the problem is not quite sure where it comes from, but it seems that thalamic circuits are not functioning properly, and by simply ablating leads small areas inside the thalamus, we can compensate for the circuitry and get them working properly. So it's kind of electric feedback problem inside the brain. So this is what is clinically done currently. People use RF ablation, where they stick an stereotactically an RF needle through the skull, drill a hole and stick it in the brain, and then ablate a small area, increase the temperature such to about 80-90 degrees, and ablate it so that the brain tissue dies, and that normalizes the circuitry. Other option is to stick a small catheter and then use it stimulated with RF signals, and that also compensates that circuitry, this deep brain stimulation. But both of these approaches are invasive, so people have tried to use also radiosurgery, but there you have to wait several months before you have the effects. And so the idea was to use this focused ultrasound for that purpose. And the targeting is simple for the thalamus. It's based on the RF experience. There are anatomical landmarks in the brain, AC and PC, that are clearly visible in the 3D scanner, and then you simply measure 7 millimeters up from PC and then 15 millimeters sideways, whichever side you want to ablate. And usually what we have done is to ablate on the dominant side of the brain, wherever the shaking is more important. And then patient is the helmet is placed into the head with pins, and then the plan is to look how the beams are propagating and take the CT information to calculate the wave distortions and then compensate for that. And then during the treatment, you can see hotspots nicely with the MRI, and you can look that they are actually, the hotspot is in right location. Typically, you compensate for a couple of millimeters at low power. So these spots are visible with a few degrees temperature elevation, so no damage to the brain, so you can get the right location. And then you just you have this movie during the treatment to show how the heating is going. So this temperature information is received about once every 3 seconds, sonication typically about 20 seconds. So here is an example of the lesion produced inside the brain, and it has worked pretty well. There are 2 studies that have been published. Elias has published the first study in New England Journal of Medicine with 15 patients, 1 year follow-up, and we had a small study about 4 patients also showing similar good results. And there have been about 80 patients totally treated with tumors and chronic pain also. And this essential tremor is now entering the phase 3 trial in 8 hospitals around the world. And this end result, what happens to the patient. So this is, the patient was asked to draw a spiral. And this before the treatment, one of our patients couldn't hold the pen on the paper. And then after going slowly with multiple sonications and reaching temperatures about 50 to 53 degrees and asking them to draw the circle again now in scanner, you can see that now they're starting to have able to draw some spirals and then a little higher temperatures, and the treatment was done, and this is after the treatment, the tremor was completely gone. So it is instantaneous gratification for the patient and for the treatment team. So our neurosurgeons have been very, very surprised and pleased with the results so far. So that's the surgery side. So that is continuing. It's in the clinic and looks very promising, at least for certain surgeries. Elisa talked yesterday about drug delivery in the brain, and the idea that we had, that now we have this focused ultrasound beam that you can put inside the brain. Can we use that to selectively disrupt the blood-brain barrier and then use the blood-brain barrier elsewhere to protect the brain and get any kind of therapeutic agents inside that image-specified location? So for us, the MR guidance has been very key to the functional guidance, the anatomical guidance for delivering agents in the brain. For this work, animal work, we have used our own animal system that goes to MR, is guided by either whole body MRI scanner or a small animal scanner, and we simply use not a phased array, but a simple spherical transducer with a hydrophone in the middle of it to listen the information coming out of microbubbles during the treatment. Our sonications are very simple. We use 10 millisecond burst once a second, about 30 to 120 seconds. Very frequency, we have tested large frequency range. It doesn't seem to have a very much impact on it. And this is a clinically relevant microbubble population. So this is the clinically accepted diagnostic agent, and we are using clinical dosages. And here are just examples in brain. This is a rabbit, rat, and mouse, showing how precisely we can aim it based on the MRI information. And here is a pig. So we wanted to, so not to repeat what Elisa was talking, but to see what additional information we have. We have done the blood-brain barrier disruptions under 2-photon microscope, where we have a small transducer ring around the microscope lens, such a way that we can image during the sonications and observe what happens on the vascular level. And here is a movie now on a sonication. Again, the sonication comes, and we have a little constriction of the vessel, and then you can see this green dye starting to leak slowly from the vessel walls. This was a relatively large vessel in this case, and the leakage was slow. We have also observed in the smaller vessels very fast leakage during the sonication, and so there seemed to be a difference. And when we look at the permeability, it looks like the small vessels are more permeable, and the leakage is fast, and then larger vessels are not as permeable, but the leakage is slow. And when you look at the vessel size here, you can see that the capillaries are the ones where you had fast leaks, and the slow leaks are mainly in the larger vessels. And when we try to understand what's going on here with the electron microscopy study, here is an example of tight junction without ultrasound, so it's the contrastation is not getting in. Here is 1 hour after sonications, where you can see this marker molecule entering the basement membrane. So it's going between the cells through the tight junction, so the tight junction is open. And then 4 hours later, you can see that now the marker molecule is entering into the tight junction, but it's already sealed here. So in our hands, the blood-brain barrier is open maximum about 6 hours, and for larger molecules, even less. So it looks like the tight junctions are healing themselves in 4 to 6 hours or so. But there was also a surprise for us, and maybe the explanation for the slow leaks, and it's here when we used the images of the vessels, we saw that there are this vacuole formation in the walls where the marker molecule is inside the endothelial cells. This is 5 minutes after the sonications, and there 60 minutes later you can see the vacuoles on the other side of the endothelial cells here and here. And so we were thinking maybe there is an active transportation that's also going on, which would be the slow leakage. And then we had a jackpot. We saw for the image where we can see one of the caveolus damping its content to the basement membrane. So there seemed to be induction of active transportation, and this is most likely happening in the larger vessels and responsible for the slow leakage. We counted the number of caveolus, and it seemed to be largest in the arterioles and much smaller in the capillaries and veins. So again, this method has been used to treat anything in the brain. Well, the first target, of course, would be a brain tumor. And people have said, okay, well, here is a glioblastoma, and you can see that there is some leakage of the blood-brain barrier already in MR contrast-enhanced scan. And people say, well, why do you need to break the blood-brain barrier? Because it's already broken in tumors. Well, this is what it looks when you do T2-weighted image, so the volume is much bigger, and when biopsies and histology has been done after patients have died, all this volume contains tumor cells in glioblastoma. So this is the target, not just this volume. So the idea is to disrupt the blood-brain barrier in this area and get the chemotherapy agents inside the brain and hopefully help the patients. Chemo is not very helpful in most cases in the brain. We did some studies with 9L gliosarcoma. Here you see the tumor. The blood-brain barrier is open a little bit after sonication. It's a nice opening around the volume. We saw tumor, this is doxorubicin delivery, doxorubicin alone, tumor doubling quite quickly, and then ultrasound doxorubicin single treatment doubling time was about twice as long as it was with the drug alone. A similar effect was observed with ultrasound alone. And then my former colleagues in Boston continued this trial with multiple treatments, 3 treatments, and here you can see surviving fraction of animals. These are the controls. They all die about 20 days. And then there is a significant increase in all of the animal survivors that were treated. And there is this about 40% of the animals which continue to survive without tumor, showing that we are getting enough drug into the brain, and it could be potentially useful in brain treatments. And we are very close to start a clinical trial using this technique now in brain tumors. We are just waiting, we have an ethics approval and waiting for Health Canada approval to start the trial. So that will be the first human trial trying to use blood-brain barrier disruption for brain treatments. Can it be used to do anything else? We have tried to put natural killer cells that are targeted in the brain tumors. We do the similar thing than before sonic gate drugs, and the idea is that we have these special natural killer cells that are targeted to HER2 specific, so we are targeting breast cancer metastases. And here you can see tumor cells. So the idea is that they get out of the bloodstream and get into the brain and then seek the tumor cells and kill them. And here is an example showing these cells actually in the bloodstream. This is after the animal is sacrificed, showing that they're propagating towards the tumor along the vessel walls. So that's a histology study we did, and we did different sonication. This is a control group without any sonication. Here is a sonication was given first, and then the cells were injected afterwards, and you can see that there are some cells inside the tumor. And then this is where we did sonication simultaneously with the injection, and you can see there are quite a few cells, and here are the numbers. So we get a reasonable number of cells in the tumor area. And we have now done a survival study and shown that there is impact on the animal survival also with this therapy. Can it be used for treating Alzheimer's disease? In Alzheimer's disease, it's characterized as plaques in the brain. And here is an example of untreated volume, and other side is a treated volume where we opened the blood-brain barrier and then gave antibody that was dissolving the clots and here plaques. And here you can see the number of plaques on the left and right side, showing that we indeed with single treatment can reduce the number of plaques on the treated side. And we had a surprise. We noticed also that by doing ultrasound treatment alone, we can reduce the plaque size on the treated volume. So it looks like just contrastation and disruption of blood-brain barrier with ultrasound does something in the brain and is able to reduce the plaque load. And we repeated this once every week for 3 weeks, and after 4 weeks sacrificed the animals, and we saw a significant reduction in the plaque load. And we had also a surprise. We noticed that there are many more new neurons in the brain of the animals that were treated. These were old, 8 months old animals that had huge plaque loads and are very old considered for any kind of therapy. And, but in this case, we had new neurons developing and plaque load reducing. So will that result in a behavioral benefit? We were surprised seeing that yes. Here are the controls. Here are non-Alzheimer's disease litter mates, and these are then the Alzheimer's disease mice. This is the control group. You can see that there is a huge reduction in their ability to recognize the novel arm. And this is the first treated group where the normal animals and the Alzheimer's disease animals are almost behaving equally. So that, and the same thing is with the alterations. So that shows that with the ultrasound treatment, without even antibodies, we can rescue the behavior potentially. So this might work in the future for treating Alzheimer's disease. And the nice thing here is that you don't need to put any other agent in, just the microbubbles and ultrasound is enough. We have also viral treatments, put stem cells in and get significant numbers in, and they seem to be surviving, and new neurons seem to be developing. So there are several options for therapy. Well, how can we do it optimally? We have to develop new devices. We are going to do first clinical trials with the current clinical device, but we need to have better monitoring than that device can provide. We have built 1,372 element array that has these empty spaces in the middle of them. So these are the transmit elements, and we have built an array of receiver elements in this array so that we can get three-dimensional map of the bubble activity in the brain. We have done experiments looking through human skull, put skull in the array, and then had microbubbles flowing through this kind of spiral tube. This is 0.3 millimeters in diameter, which is about the smallest size you can see with CT and through human skull or MR. And then we did experiments. It was also in one experiment did put a rat and see if you can see the bubbles when you open the blood-brain barrier. And here is what we were able to reconstruct of that spiral. We can see it well with the normal resolution. And then we used super resolution techniques borrowed from optics, and you can see that we can localize these microbubbles way beyond the diffraction limit and see individual bubbles. So we should be able to get very nice control on the bubble level of these blood-brain barrier disruptions in patients. So this is true human skull imaging. So and the scale is here is 1 millimeter. So we should be able to do this in a human. Of course, more work needs to be done, but here is an example of a rat, how we can see the location where we are having the bubble activity, and we can separate if we produce tissue damage and not only blood-brain barrier disruption. So as a summary, I came with quite a few lots of results, but MR-guided focused ultrasound can be used safely in a clinic to have focal thermal coagulation of the brain tissue, and this is entering now in phase 3 trials. The phase 3 trial has actually started already. And MR-guided focused ultrasound can induce transient, safe, and local blood-brain barrier opening for drug delivery and also cellular therapy. And many molecules and cells can be can reach the brain at the quantities which are adequate for therapy or diagnosis of the brain. And this technology is ready to enter clinical testing. And thank you very much.
Questions?
Kullervo, in this clinical work with essential tremor, the explorers are done with compensation for skull intragenetics the same way you did it?
Yeah. So it's using the algorithms that we developed originally, but the company has, I don't know how they modified them, but that's the basis for it.
And for imaging, you also use?
So for what we ISO at the end, yeah. So that was done compensation both ways, transmitting and receiving.
So now MR will result.
The last one was no MR.
Okay. So,
in the talk about the GBM, where you're delivering or bringing blood-brain barrier for the chemo, would that have to be done before each chemo treatment? If they're doing, say, 5 days of treatment, would you have to reopen the blood-brain barrier each day?
the request is to repeat the question, to be honest, in the back.
Yeah, so the question is that if this is used for chemotherapy treatments, does it need to be repeated for each treatment? Yes, the answer is yes. So.
Is there any additional considerations if you're doing this on, say, a daily basis or a week?
So we have done some treatments in animals daily basis, and it seemed to be doable. Of course, it gets with MR, it gets pretty expensive. So for that kind of treatments, maybe there are other things we have to do. But in the animals, it looks like once a week, 3 treatments was enough. So it's probably not that extensive.
Could you say something more about the super resolution imaging approach?
Yeah, so what we are doing is we are using the array, or receiver array, to reconstruct the beam paths, making compensations for the skull propagation, so we can localize the bubble information and then use the optical Gaussian fitting techniques to have a super resolution of the location of the bubble, so using low concentrations.
Yeah, I have more questions about the array that you were using to do that. Can you describe that in a little more detail?
So the transmit array is 300 kilohertz array. It's made of PCT tubes and it's using lateral coupling, so the electrodes are in the tube walls, so we can get the impedance right. And it's 1,300 plus elements. And then we have the receiver elements, PCT elements in the middle of those tubes. We used 128 elements, a sparse array, randomized or optimized sparse array actually. And then those were tuned to the second harmonic frequency.